Okay, so get this.
Okay.
Imagine like a really common drug,
Mm.
the kind you take for like a simple parasite.
Mm-hmm.
And then suddenly it turns into a secret weapon to fight ovarian cancer.
Wow.
Crazy, right? It's like
Yeah.
something out of science fiction.
Yeah, totally.
Well, that's what we're diving into today.
Sounds intriguing.
You sent over this research paper called Research Perspective, Potential Role of Nitazoxanide in Ovarian Cancer Treatment. Old drug, new purpose.
Yeah.
I mean, the title alone had me hooked.
Yeah, it's a fascinating concept.
So let's break this down.
Okay, sure.
But first, maybe we should start with the bad news first.
Yeah, that's a good point.
Like, why is ovarian cancer so serious?
Well, ovarian cancer is unfortunately a very aggressive disease.
Yeah.
It's actually the leading cause of death from gynecologic cancers.
Wow.
And that's largely due to the high recurrence rates.
Oh, gosh.
And the limited treatment options that we have, especially for those with advanced disease.
Yeah, that's what makes this research so important. What if this drug, Nitazoxanide, or NTZ as I think we'll call it,
Sure.
what if it could actually offer like a new way to fight this?
It's a very exciting possibility.
But before we get too far ahead of ourselves,
Right.
can you kind of back up and give us the big picture?
Sure.
Like, what makes treating ovarian cancer so difficult in the first place?
Well, one of the biggest challenges is drug resistance.
Drug resistance.
Cancer cells are unfortunately very adaptable.
Oh, yeah.
They evolve over time, and they can actually evade the treatments that are designed to destroy them.
So they're basically outsmarting us.
Yeah, in a way, you could say that.
Wow.
And that's why we're always looking for these new strategies.
Yeah, to stay one step ahead.
Exactly.
Okay, so where does NTZ fit into all of this?
Okay.
I know it's usually prescribed for parasites, right?
That's right.
Not for cancer.
It's typically used for treating certain parasitic infections, and it's actually a very safe and inexpensive drug.
Oh, wow.
It's already FDA approved.
So it's been around for a while.
Yes, but what's really remarkable about this drug is it's incredibly versatile.
Versatile.
It has other uses.
Okay, so it's got a few, hidden talents.
Yes, you could say that.
Okay, I'm intrigued. Lay it on me.
So in addition to its anti-parasitic activity, it's also shown some antiviral effects against hepatitis C.
Really?
And it can even stimulate autophagy, which is a process we'll talk about more later.
We'll definitely have to dive into that.
But one of the most fascinating things is that it seems to have this potential to inhibit a protein called protein disulfide isomerase, or PDI.
Okay, PDI.
PDI.
I'm not familiar with that one.
So
What is it, and why is it important when we're talking about cancer?
okay, so think of proteins like these really intricate origami creations.
Okay, I like that analogy.
To function correctly, they need to be folded in very precise ways.
So they have a specific shape.
Exactly, and PDI is kind of like this tiny little folding assistant.
The folding is it.
It makes sure that all these proteins get their shapes right, especially within a part of the cell called the endoplasmic reticulum, which is kind of like the protein factory of the cell.
Okay, so PDI is making sure the protein origami is done correctly.
Precisely.
Okay, I get it. But how does this relate to cancer?
Well, it turns out that cancer cells, especially in those harsh environments that tumors create, they really rely on PDI.
They needed to survive.
Exactly. It's like PDI is part of their survival toolkit, helps them cope with all the stress.
So it's like their secret weapon.
In a way, yeah.
Okay, so if we could somehow mess with PDI, stop it from doing its job,
Yeah.
could that, disrupt the whole protein folding thing and make those cancer cells weaker?
That's the idea, and that's what's so exciting about this research.
Right.
So inhibiting PDI could trigger something called ER stress.
ER stress.
And that basically overwhelms the cell's ability to handle these misfolded proteins.
Oh, wow, so it's like a system overload.
Right, and this can potentially lead to cell death, especially through this process called autophagy.
Okay, hold on. We've got a lot of moving pieces here. We're talking about a parasite drug,
Right.
a protein folding assistant, cellular stress, and now autophagy.
Yeah, it's a lot to keep track of.
Can you connect the dots for me?
Sure.
How does it all fit together, and where does NTZ come into play?
So the researchers who are working on this paper, they noticed that NTZ actually shares some similarities with another group of molecules called PATM, which have shown some anti-cancer activity by inhibiting PDI.
So they're like cousins of NTZ.
In a way, yeah, and that's what sparked their curiosity.
So they thought, hey, if it can fight parasites, maybe it can fight cancer cells too.
Exactly. It's that kind of outside-the-box thinking that can lead to these breakthroughs.
I love it.
And it seems like their hunch is paying off.
Early
studies suggest that NTZ, by inhibiting PDI, could actually trigger this whole cascade of events in ovarian cancer cells. ER stress,
Uh-huh.
leading to cell death through autophagy.
This is amazing.
It's very promising.
Okay, but I need you to walk me through this autophagy thing.
Okay, sure.
Break it down for me like I'm 5 years old. What's happening inside these cells?
Okay, so imagine that your cells have this built-in recycling system.
Okay.
Autophagy is essentially that system in action.
Okay.
It's how cells get rid of damaged parts, worn-out organelles, even invading pathogens.
Well, it's like a cleanup crew.
Exactly. It's a constant process. It's like taking out the trash and composting, you know? It's essential for keeping things running smoothly.
So autophagy is keeping the cells nice and tidy.
Precisely.
But didn't you say something about autophagy having like 2 sides?
Yeah, that's right.
So it's not always a good thing?
Well, at low levels, autophagy is actually protective.
Protective.
Yeah, it helps cells adapt to stress and recycle components.
Okay.
But if the stress is too severe or if it goes on for too long, it can become a problem.
So it's like pushing that recycling system into overdrive.
Yeah, exactly.
And then the cell just can't keep up.
Right. It's like the cell is digesting itself from the inside out.
Wow, that's intense.
It is.
But if we're talking about getting rid of cancer cells, maybe that's not such a bad thing, right?
You're exactly right.
So we want autophagy to go into overdrive in cancer cells.
That's the idea. That's the exciting potential here.
Okay, I'm following you.
If we can selectively trigger this really intense destructive form of autophagy in cancer cells, it could be a powerful new way to treat cancer.
And this is where NTZ comes back into the picture.
Exactly.
So by inhibiting PDI and causing ER stress,
Yes.
it's basically pushing those cancer cells toward this self-destruct button.
That's the hypothesis. And what's really encouraging is that NTZ is already approved for human use.
Right.
So we already know a lot about its safety profile.
So they could speed things up.
Yes, it could potentially really fast-track its development as a cancer treatment.
This is so cool. Instead of starting from scratch, we're just repurposing a drug that's already out there.
It's a very efficient approach.
Talk about efficiency.
And this whole old drug new purpose approach is gaining a lot of traction these days.
Yeah, I've been hearing about it.
Researchers are really starting to look at all these existing medications.
For new applications.
Exactly. It's like we're raiding the medicine cabinet looking for these hidden treasures.
That's a great way to put it.
But, you know, we have to emphasize that this research is still very early.
Right.
The preclinical data is promising.
Mm-hmm.
But we really need to conduct these rigorous clinical trials.
Yeah, we need like the human data.
To confirm that NTZ is both effective and safe for people.
Right. We don't want to get ahead of ourselves.
Exactly.
But even if NTZ doesn't end up being the magic bullet for ovarian cancer,
Yeah.
what we're learning about these cellular processes
Right.
that could have implications beyond just this one type of cancer.
Absolutely. This research really highlights the importance of understanding these basic mechanisms that drive how cancer cells behave and survive.
Yeah, if we can understand them,
Yes.
then we can outsmart them.
Exactly. The more we learn about these processes like ER stress and autophagy, the better equipped we'll be to develop these really effective treatments.
Not just for ovarian cancer, but for other types of cancer too.
Exactly.
So this deep dive is like taking us way beyond just one drug and one disease.
Yeah, it's much bigger than that.
It's like changing how we think about cancer treatment.
It is.
It's not just about finding like a magic bullet.
Right.
It's about understanding the enemy from the inside out.
That's a great way to put it. We're looking for ways to exploit the vulnerabilities of cancer cells.
Turn their own defenses against them.
Exactly.
Which brings us back to this fascinating idea of autophagy.
Right.
It's like this ancient cellular process that's essential for life.
Uh-huh.
But it has this dark side that we might be able to use to our advantage.
It's a perfect example of how nature often provides solutions in the most unexpected places.
Yeah, it's so cool that we went down this rabbit hole today.
I agree.
But before we get too carried away thinking about the possibilities, why don't we take a step back and summarize what we've learned so far?
That's a great idea.
So we started by looking at those grim statistics of ovarian cancer,
Yeah, those are tough.
talked about the challenges of drug resistance,
Right.
and the urgent need for new treatment options.
We definitely need new options.
And then we went deep into the world of PDI,
Yeah, that tiny little protein folding assistant.
the one that cancer cells can't live without.
Exactly. And it seems like inhibiting PDI is a really promising strategy.
Yeah, and that's where NTZ enters the scene.
Exactly.
We talked about how NTZ, this drug that's typically used for parasitic infections, might also be able to inhibit PDI.
It's very promising.
And that could trigger this whole chain reaction.
Yes.
That leads to ER stress and eventually cell death through autophagy.
It's a fascinating cascade of events.
It really is. And we also spent some time unpacking all the complexities of autophagy.
Right.
It's such a fascinating cellular recycling system
It is.
that can either help or hurt the cell depending on the situation.
It's a beautiful example of how delicate that balance of life is at a cellular level.
It really is. Okay, so we've covered a lot of ground today.
We have.
What does this all mean for our listener?
That's a good question.
Like, why should they care about this research?
Oh, for one thing, it offers a real glimmer of hope
Yeah.
in the fight against this really challenging cancer.
Mm-hmm.
You know, if NTZ proves to be effective in clinical trials,
Right.
it could be a really valuable addition to our arsenal of weapons against ovarian cancer.
Yeah. Fingers crossed.
And it also highlights the potential of this whole drug repurposing approach.
Right.
You know, finding new uses for existing medications.
Yeah, we talked about that.
It's a really exciting avenue of research.
But it's not just about ovarian cancer.
No.
This research teaches us about these really complex cellular processes like ER stress and autophagy.
Uh-huh.
And these processes, they're involved in so many different diseases.
They are.
So the more we learn about them,
Yeah.
the better we'll be at developing effective treatments. And finally, I think this research really speaks to the power of scientific inquiry
Absolutely.
and the importance of thinking outside the box.
Yeah. Sometimes the most unexpected solutions are hiding in plain sight.
This whole deep dive has been a perfect example of that.
It really has.
It's not just about one drug or one disease.
Right.
It's about a whole new way of thinking.
It is.
A new way of approaching problems.
And it reminds us that we all have a role to play in advancing scientific progress.
Yeah.
Whether we're researchers or clinicians, patients, or just curious individuals, we can all contribute to this quest for knowledge.
Yeah.
And ultimately, better health outcomes for everyone.
I'm so glad we had this conversation today.
Me too.
But we're not quite done yet.
Oh.
I have one final thought-provoking question for our listener to ponder as we wrap up.
Okay, I'm ready. Okay, so picking up where we left off, let's zoom in on this whole autophagy thing.
Okay.
So, you know, it's more than just this, like, cellular cleanup crew. It's really this, like, sophisticated recycling system with, like, a touch of macabre elegance.
Okay, more than just taking out the trash.
Yeah, think of it as a cellular composting operation.
Okay, I'm listening.
So when a cell senses, like, damaged components or, like, worn-out organelles or even, like, invading pathogens.
Bacteria and viruses and stuff?
Exactly.
Oh, okay.
It basically wraps them up in these, like, double membrane vesicles.
Vesicles?
It's called autophagosomes.
Autophagosomes.
Yeah, it's a mouthful.
Okay, so like tiny little biohazard bags for the bad stuff.
And then these autophagosomes, they fuse with another cellular compartment called the lysosome.
The lysosome?
Yeah, have you heard of that before?
I have heard of that, but remind me what happens there.
Okay, so the lysosome is like the cell's recycling center.
Okay.
It's packed with all these powerful enzymes.
So it's where the recycling magic happens.
Exactly. It breaks down all that cellular waste into these reusable components.
So it's not just getting rid of the garbage, it's like breaking it down and then using the parts for something else.
Exactly. It's incredibly efficient. The broken down components are then used to build new structures or to provide energy for the cell.
Okay.
And this process is constantly happening at a basal level in all of our cells.
Just keeping everything running smoothly.
Exactly.
Okay, so autophagy is like this ongoing maintenance crew, you know, keeping the cell in tip-top shape.
Yes.
But we were talking about how this helpful housekeeper can turn into like a Terminator.
Yeah, it's true.
How does that happen? What flips the switch?
Well, it all comes down to like the intensity and the duration of the stress.
Okay.
Think of it like exercise.
Yeah.
You know, a moderate workout is good for you. It builds strength and resilience.
Right.
But if you push yourself too hard for too long...
Oh, you get injured.
Exactly. You can damage your muscles. So it's kind of the same thing with cells.
So if the stress is too much, autophagy goes from like a gentle jog to a full-on sprint.
Right.
And the cell just can't handle it.
Exactly. The recycling process goes into overdrive.
Okay.
It starts breaking down these essential components faster than the cell can repair or replace them.
So it's like the cell is eating itself.
Yeah, it's like the cell is digesting itself from the inside out.
Okay, that's a little creepy, but if we're talking about cancer cells, maybe that's what we want.
Yeah, right. Exactly. That's the exciting potential here. If we can selectively trigger this really intense, destructive form of autophagy in cancer cells, it could be a completely new way to treat cancer.
And this is where NTZ comes back into the story.
Right.
So by inhibiting PDI and causing that ER stress, it's like pushing those cancer cells toward this like self-destruct sequence.
That's the hypothesis, and what's really great is that NTZ is already approved for human use.
Right.
So we already know a lot about its safety profile, which means we could potentially fast-track its development as a cancer treatment.
So we're not starting from scratch.
Exactly.
We're kind of like repurposing an existing drug, which could save a lot of time and money.
Absolutely. It's a much more efficient approach.
And you said that this whole old drug, new purpose idea is gaining a lot of momentum.
Oh, it is. Researchers are increasingly looking at existing medications for new applications.
It's like we're reading the medicine cabinet for hidden treasures.
I love that analogy.
Me too. But, we have to be realistic here.
Of course.
This research is still in its early stages.
Very early.
The preclinical data looks really promising.
It does.
But we really need those rigorous clinical trials to confirm that NTZ is both effective and safe in humans.
We can't skip that step.
Right. We don't want to get ahead of ourselves.
No, we need to see how it actually works in people.
Okay, I get it.
Yeah.
But even if NTZ doesn't turn out to be the answer for ovarian cancer,
Yeah.
what we're learning about all these cellular processes
Right.
that could have bigger implications.
Absolutely. This research is so important because it highlights how important it is to understand these fundamental mechanisms that drive cancer cell behavior and how they survive.
Yeah, if we can understand them,
Yes.
then we can figure out how to beat them.
Exactly. The more we learn about things like ER stress and autophagy, the better we'll be at developing these really effective treatments.
And not just for ovarian cancer.
No, not just for ovarian cancer, for other types of cancer as well.
Okay, so this deep dive is taking us way beyond just one drug and one disease.
It really is. It's about a much broader shift in how we approach cancer treatment in general.
It's like a whole new way of thinking.
It is.
We're not just looking for that magic bullet anymore.
Right. We're trying to understand the enemy from the inside out.
We're getting up close and personal with the cancer cells.
Exactly. We're looking for ways to use their own defenses against them.
Which brings us back to this whole fascinating concept of autophagy.
Yes.
It's this ancient cellular process that's essential for life,
Uh-huh.
but it also has this, dark side
Yeah.
that we might be able to harness for good.
It's a beautiful example of how nature often provides us with these solutions in the most unexpected places.
I'm so glad we went down this rabbit hole today.
Me too.
But before we get too carried away with all the possibilities,
Okay.
why don't we take a step back and summarize everything we've learned so far?
That's a good idea.
So we started by looking at those really grim statistics of ovarian cancer.
Yes. Those are tough numbers.
But talked about drug resistance.
Yeah.
And how badly we need new treatment approaches.
Absolutely.
Then we dove into this whole world of PDI.
That tiny protein folding assistant.
That cancer cells just can't seem to live without.
Right.
And it seems like inhibiting PDI could be a really promising strategy.
It could be a real game changer.
And that's where NTZ comes in.
Exactly.
This drug that we typically use for parasitic infections.
Mm-hmm. Might have this really unexpected ability to inhibit PDI.
Yeah, and that could trigger this whole chain of events.
Yeah, this fascinating cascade of events.
Leading to ER stress and eventually cell death through autophagy.
It's amazing how everything is connected.
And we spend some time really breaking down the complexities of autophagy.
Yes.
It's such a fascinating cellular recycling system.
It is.
That can be both a friend and a foe, depending on the circumstances.
It really is a delicate balance.
It's incredible how life works at a cellular level.
It is.
So we've covered a lot of ground.
We have.
But I want to bring it back to our listener.
Okay.
Why should they care about this research?
Well, for one thing, it offers a real glimmer of hope in the fight against this very challenging cancer.
It's a big deal.
If NTZ proves to be effective, it could be a really valuable new tool in our fight against ovarian cancer.
Fingers crossed.
And it also highlights the potential of this whole drug repurposing approach.
Right. Using old drugs for new tricks.
Exactly. And finding new uses for medications that are already out there.
Yeah. And it's not just about ovarian cancer.
No, it's not.
This research really sheds light on all these complex cellular processes like ER, stress, and autophagy. And these processes are involved in so many different diseases.
They are.
So the more we understand them, the better equipped we'll be to develop effective treatments for all sorts of things.
Absolutely.
And finally, I think this research speaks to the power of scientific inquiry.
I agree.
And the importance of thinking outside the box.
Yeah, sometimes the most unexpected solutions are hiding in plain sight.
This entire deep dive has been a perfect example of that.
It really has.
It's not just about one drug or one disease.
Right. It's about a new way of thinking.
A new way of approaching problems.
And it reminds us that we all have a role to play in advancing scientific progress.
Yeah.
Whether we're researchers or clinicians or patients or just curious individuals, we can all contribute to this quest for knowledge.
And hopefully,
yes,
better health outcomes for everyone.
I couldn't agree more. This has been a really great discussion.
I'm so glad we had this conversation today, but we're not quite done yet.
No.
I have one final thought-provoking question for our listener to ponder as we wrap up.
Okay, I'm all ears.
Could this whole old drug thing, like, change how we approach cancer treatment?
It's a really interesting question.
Like, could it lead us to explore other drugs that are already out there,
Yeah.
for new purposes?
It's definitely a possibility, and it has a lot of researchers really excited.
It's like a whole new frontier in medicine.
Yeah, it really is.
I see why.
I mean, if we can figure out new ways to use these drugs that are already considered safe and effective
Right.
for other conditions, it could potentially save us so much time and money.
Years of research and development.
Exactly. Not to mention, it could make new treatments more accessible and affordable for everyone.
Yeah, that's huge.
It's a huge deal.
Think about how much it costs to bring a brand new drug to market.
Oh my gosh, it's a fortune.
It's a long and complicated process.
Incredibly complex.
But with drug repurposing, it's like we're skipping over a lot of those steps.
Yeah, we're basically fast-forwarding through all the red tape.
We're like leapfrogging over those hurdles.
Exactly. And because we already know so much about these drugs,
Yeah.
their safety, their mechanisms of action, their potential side effects,
We're not starting from scratch.
we're not. And that could be a real game changer in areas like cancer research where we really need new and innovative therapies.
Absolutely.
It makes you wonder what other hidden gems are out there just waiting to be discovered.
Right.
Drugs that we might be using for everyday things,
Yeah.
could actually hold the key to these breakthroughs for cancer or other diseases.
It's pretty amazing.
It's the thrill of scientific exploration. We're always learning, we're always pushing the boundaries of what we know. And sometimes the most groundbreaking discoveries, they come from the most unexpected places.
Yeah.
It's all about connecting the dots, seeing things in a new light.
And this whole deep dive has been a perfect example of that.
I agree.
It's not just about one drug or one disease. It's a new way of thinking, a new way of solving problems.
And it's a good reminder that we all have a role to play in moving science forward.
Absolutely.
Whether you're a researcher or a clinician, a patient, or just someone who's curious about the world, we can all contribute to this search for knowledge and ultimately better health for everyone.
That's a great message to end on.
I think so.
So to our listeners out there, if you're intrigued by what you've heard today, keep asking those questions.
Yes.
Keep exploring. You never know what you might find.
Sometimes the most unexpected paths lead to the greatest discoveries. So until next time, keep diving deep.